Thank you very much. I'd like to call our panelists to come on up and take your seats. And while you're doing that, I'm going to just talk for a little bit. In 1996, I returned from Japan having launched Intron A for Hep C, and one of the first papers I wrote was the way we treat hepatitis C is going to change. I had launched coverage on about 14 different companies talking about a host of antivirals. Could be protease inhibitors looked like the winners, where you might see non-nucleosides the winners. It wasn't clear how that industry was going to shake out. Fast forward 20 years, and we see that Gilead looks like they've virtually won the race with an STR. Turns out that it's a polymerase inhibitor and the combination of an NS5A. What's amazing about Gilead's drug is they shattered all records for the most robust launch in the history of pharmaceuticals. When Gilead acquired Pharmasset for $11 billion, it was one of the largest acquisitions we'd ever seen, and it looks like that acquisition will be completely paid for within the second year of its launch. So paradigms shift. They really do. 10 years ago, I started getting interested in cell therapy. And, we've been watching the entire space, autologous, allogeneic, pluripotent, mesenchymal. And 10 years ago, I really had no idea where this space was going. As a research analyst, I can tell you we're seeing a very similar dynamic to what I always see. History repeats itself. The industry is starting to sort itself out. It's becoming more clear where the winners are going to be. And what's driving that process? Data. Understanding the data and understanding the catalysts. So it's a very exciting panel here today because what we're doing is we're not just looking at regenerative medicine, my area of expertise, but we're also looking at gene therapy. Gene therapy is relatively complex. It's not an area that I understand that well, so I'm so pleased that my colleague, Dr. Jason McCarthy, on the other end of the panel, will talk a little bit about those companies and where they're going. And I'm very excited about our last speaker, who really represents both sides of that coin, regenerative medicine and gene therapy. I've asked all of the panelists to focus on two things. Let's keep it simple. What catalysts do you have over the next year so that I should believe your stock is worth owning? How meaningful will those catalysts be? What's that data going to look like? Will there really be proof of concept in that data? Can I look at your data and can I have a high level of confidence that there's a drug here and that this is going to work? And what's really exciting for me on the regen side is that I have Mesoblast and Athersys. Because Mesoblast and Athersys have both had a lot of news in the last week. Dr. Gil van Bokkelen probably just came back from Japan recently and has announced a very significant partnership with Helios. And I'm going to ask him to talk a little bit about what that partnership is and what it means. But more importantly, Dr. Itescu is making me work this morning at 5 o'clock announcing data that the plans for the congestive heart failure change have shifted a little bit. Looks like they've gotten a little bit better because the powering calculations, the amount of patients enrolled, and the primary endpoint of that trial has shifted. And what's really exciting for me is, and Edward, I saw the slide and I saw how prominent cancer is, but when we talk about cardiovascular medicine and we talk about stroke, the size of those market opportunities are just off the scale. They really are unmet medical needs. So if a company like Mesoblast can deliver definitive proof of concept, already partnered with a global pharmaceutical giant like Teva, what does that mean? And so nobody should be distracted by the price of Mesoblast today. No one should be distracted by where Athersys is today. Because where Athersys is today really has no bearing on where it's going to be if they produce meaningful data. And those are the topics we want to explore. Thank you very much. I look forward to Dr. Itescu talking to us a little bit about the Mesoblast news that came out this morning.
Thanks very much, Jason.
You're welcome.
So what we announced this morning was that in our phase 3 trial in chronic heart failure, which had previously been designed as a time to first event analysis, a one-to-one randomization comparing outcomes between placebo treated patients and patients who received a single catheter-based injection into the left ventricle. The previous number of patients was about 1,165 patients. We've had very, very good discussions. Actually, our partner Teva had a very positive discussion with the FDA, who agreed to a modification of the endpoint from a time to first event analysis to a multi-event analysis, comparing again, one-to-one randomized placebo versus cell-treated, where the outcome is not a first heart failure hospitalization, but really a reduction in overall hospitalizations and recurrent events. Now, recurrent events is a much more important clinical endpoint than a first event, of course, because most patients with advanced heart failure don't have a single hospitalization, but rather have recurrent events and then die. So the pharmacoeconomic burden is really at the level of repeated hospitalizations, and if you can prevent multiple hospitalizations, you're doing a great service for the healthcare community, as well as obviously optimizing the potential for reimbursement. And I guess what I'm particularly happy about was how embracing the FDA was of this change in the primary endpoint, where they understood the unmet need and they understood the data that we've generated in phase 2, because in our phase 2 trial, what we really showed was not only that we prevented a first hospitalization relative to the placebo patients, but we prevented multiple hospitalizations, actually 0 events in the cell-treated versus 11 hospitalizations in the placebo patients. And in fact, the predictability of preventing these events was seen early on, when we could prevent the diastolic and systolic dysfunction in these patients, which then allowed us to project 2 or 3 years out. And in fact, the optimal treatment benefit was seen in those patients who had the most advanced forms of heart failure, which again allowed us to have a very meaningful discussion with the regulator to say that in fact this is a terrible unmet need. It's the most advanced forms of heart failure, those patients with the most dilated cardiomyopathy, where if the technology works, we're going to have a major impact, and where there are no alternative therapies, and certainly no standard pharmacotherapeutic approaches.
Silviu, help me understand how the transition from a single event to multiple events impacted the powering calculation so that you could drop the enrollment of the trial from, I believe it was 1,100 and change, to now, I think, 600 and change. And also, I'd just like you to mention, too, I know there's a second trial coming. Help me understand the timing of that second trial, too.
Sure. So effectively, if you look at this in a simplistic way, a traditional time to first event analysis requires a single patient to meet an outcome, and then they get censored. And so you need a lot of patients to meet your desired event size and treatment benefit between placebo and treatment. If you're looking at a repeated analysis of repeated events, then you're more likely to see those events with a smaller number of patients, when in fact the disease results in repeated hospitalizations and events. So by having the discourse with the FDA to say that in fact the disease burden here is what we should really be trying to reverse, and not just a first event, I think the reason for a reduction in patient size from 1,100 to 600 is simply an acknowledgement that for every patient there's at least 2 events. That means that we can recruit half as many patients effectively without losing any powering, and without giving up taking any statistical hits.
Good, and we'll come back to the second trial, but I also want to throw in And you made that very clear, by the way. So 2 events per patient cuts the powering, or allows you to maintain the powering, but cut the N value in half, essentially.
So that means that the trial will be faster to recruit, faster to complete, and it will be less expensive. And all of that is, of course, a win-win for both ourselves and our partner.
Oh, so here's the quintessential question, which is, at what, I believe there is a second interim analysis. So given, and you know a little bit about what the enrollment velocity looks like today, do you have any idea when we might see that second interim, and how meaningful will that interim analysis be? And then how, this is really, really important, I think. Where is Teva in all of this?
Those are very, very broad questions, but I think in the first instance, we have a first interim analysis that is going to read out towards the end of this quarter, beginning of the second quarter, and that's roughly on the first 120 patients who've been followed for about 6 months, looking at surrogates of the primary event, surrogates being systolic and diastolic volume. And we, as I said, we saw very, very dramatic effects in the phase 2 trial. So if we see the similar directionality in the first interim, then I think we'll be very pleased in terms of the administrative look. It's important to note that in this first interim, we're not looking at the primary endpoint. There will be a second interim analysis that is roughly when about 50% of total events have occurred. So we would expect that second interim to occur towards first quarter of next year. That is a futility analysis. It looks at the primary endpoint of heart failure hospitalization or death, and inbuilt in there is a test for overwhelming superiority, which, if we're lucky enough to hit, obviously would potentially allow us to have an early stop.
So what I hear you saying is that the first interim analysis now actually can move a little bit beyond just safety, when, I think that safety is going to be okay, but now we're actually going to get a read on efficacy through a surrogate marker like end systolic volume.
That's correct. Okay. And that 6-month end systolic volume and diastolic volume has a correlative association with reduction in hospitalizations downstream.
So, you've been involved with Teva for many years, and there have been a lot of changes in Teva's management. Help me understand, Teva seems to be talking a little bit more about Mesoblast than they have before. Where is Teva in all of this with you?
Look, I think Teva is a company that has come from the generic space to now being involved in a number of branded areas. Their primary area of focus remains CNS, of course, and clearly cardiovascular is not their principal area of focus. Nonetheless, they've been a terrific partner over the past 18 months or so. They've driven this program very hard. They've led strategically the discussions with the FDA and with EMA. And in fact, the program is now moving beyond the US into Europe. And I think Teva has shown a very conservative but appropriate approach to building this program. And I think what you're going to see is, with continued positive clinical data, you'll see even greater engagement and greater resources put behind the program by Teva.
This morning we're talking about congestive heart failure and Mesoblast. We're not talking about some of the other programs, but I think we're doing that because CHF is so large and it's so clinically meaningful. But for me as an analyst, I also see proof of concept as resonating from all of the other, very exciting areas that you're involved with, like GVHD and degenerative disc disease. With that, let me ask you to turn the microphone over to Dr. Gil van Bokkelen. Gil, thank you so much. You know that I'm very excited about what Athersys is doing in stroke, and the idea of being able to extend the golden window from that, 3 to 6 hours to 24 to 30 hours is really, really exciting. There was a time on Wall Street, by the way, when phase 2 clinical trials were really meant to figure out how the drug works and not just to have clinically meaningful data and definitive proof of concept. And I see Dr. McCarthy chuckling because we're almost at the point on gene therapy where we expect N to equal 1 in a phase 1 clinical trial to tell you that the drug works. And there's a little bit maybe of irrationality in all of that. A lot of people were trying to understand the meaning of the phase 2 data, trying to understand the meaning that you didn't move forward with Chugai. But last week you announced a very exciting partnership with Helios, which seems to me 15 million reasons to believe that the post hoc analysis really does show the seeds of what could be a very definitive phase 2 clinical trial. Can you give us an update on where is Athersys in multistem, in stroke, and in Japan?
Yeah, absolutely. So first off, thanks for having me as part of the panel today. We were thrilled last Friday to announce the new partnership with Helios in Japan, and we are incredibly excited, and I know I speak for our new partner as well, about their enthusiasm and passion for this program and what it could mean, particularly in the context of the new regenerative medicine framework, the regulatory framework in Japan. And Silviu, you didn't talk about it, but you guys actually had a recent victory on that front as well through the partnership with JCR in terms of not only getting full approval under that new framework, but also getting a very, very attractive reimbursement framework, which sets a precedent, really, I think, for the rest of the field. So congrats on that, along with the other successes. We in Helios looked at this, and you know, you mentioned Chugai, Jason. So first off, let me just say, we have the utmost respect for Chugai as a partner, and it was a very difficult decision for us to transition away from Chugai into the new partnership, but we felt it was the right thing to do for multiple different reasons. And I think that some of the things that we've learned from the data that has emerged from the stroke clinical program over the past few months, and some of the upcoming data that will be presented, for example, at the International Stroke Conference in Los Angeles in February, and then other presentations that we make along the way, I think really give a vivid understanding and illustration of what it might mean in terms of this data being so impactful and being able to extend that treatment window, Jason, which you referred to. The data shows in a very compelling way that if we treat patients within a 36-hour window, we see very dramatic, very robust improvement in these patients that translates directly to the types of things that clinicians want to see, that obviously our partner wanted to see. And I think it really speaks to being able to address an area that in some ways kind of represents one of the holy grails, if you will, in terms of clinical medicine, being able to address a substantial area of unmet medical need. In Japan, you're talking about several 100,000 patients a year that suffer a first-time ischemic stroke. Globally, you're talking about somewhere between 15 and 16 million patients that suffer a first-time ischemic stroke. And just in the three geographies we care most about, in the European Union, the US, or North America, and in Japan, you're talking about 2.2 million first-time ischemic stroke patients a year. And it's important to recognize that only less than 10% of those patients currently get to the hospital in time to get treated with TPA, which has to be administered within the first several hours, or to undergo a mechanical thrombectomy procedure. So you're talking about a very small number of patients, and now you're talking about data that is, we believe, and our new partner believes, and I think a lot of clinicians that have seen the data certainly believe, that this data could represent a quantum leap forward in terms of allowing us to reshape how we think about stroke care and being able to practice that and implement that in a clinically practical, very scalable kind of way. So it's for all those reasons, the magnitude of the opportunity, I think the robustness of the data, the new regenerative medicine framework in Japan, that we and our partner Helios are so excited about this opportunity and driving it forward. One of the questions that you asked, Jason, was what are some of the catalysts that are coming up in the course of the year? Well, obviously we'll be presenting more information on the stroke program. We've now completed the last subject one-year follow-up visit for that, so we'll have the opportunity to talk about that in due course. I mentioned that we expect at least a couple of presentations at the International Stroke Conference in Los Angeles in late February. We've got two other active clinical programs, one in the acute respiratory distress area, which is actually a part of the design, if you will, of our partnership with Helios as well. It's an area that we could expand into together because it's a serious problem. Again, great unmet medical need, both in Japan as well as here in the United States and other parts of the world. And we got an active phase 2 clinical program at treating patients that have suffered myocardial infarction. So we're expecting data to kind of roll out and progress from each of these programs over time, and I think those programs plus the other things that we've been very active in, plus the other ongoing partnering discussions that we're very active in, I think really give us a lot of reasons for optimism and certainly a lot of excitement headed into this year.
Gil, you talked a little bit in the press release about the stroke trial possibly initiating in first half of this year. Can you give us some idea of what you're thinking? Would that be a global trial? Will it be just a Japan trial?
Yeah.
How big might it be? Would there be an interim look? What we really want to know, I want to come back to this question of, at what point might we start to see proof of concept that we've got a therapy that works for stroke?
Yeah, so we actually disclosed in the press release that we're targeting the initiation of the study in Japan in the second half of the year, and then in parallel with that, we're trying to be a little bit conservative because I can tell you one of the nice things, we in Helios and Hardy, who's sitting here down in the front, we didn't waste any time. We actually went in and had our first meeting with PMDA even before we announced the partnership. And I think that's a testament to how aggressively we're thinking about this, how quickly we're moving together. There's just incredible synergy and strength in terms of the chemistry between the teams at the organizations, and we've really hit the ground running, which is something that I'm really, really excited about, and I think that bodes well. There's such a consistency of the vision and just compelling commitment between the two organizations to work together to get this done as quickly and efficiently as possible. So we're moving aggressively at getting this first study up and running in Japan, but in parallel with that, we're working on launching the international study in the US, North America, and also the European Union so that we can actually advance these two programs together. The first priority, as I'll talk about in my presentation later today, and as Hardy talks about in his presentation on Wednesday afternoon, is really going to be about getting the study in Japan up and running as quickly as possible, and then slightly staggered behind that, the international study. We haven't disclosed a precise timetable of when we expect to have data, and I certainly, Hardy will be adding some additional color at his presentation later on in the week. But let me just say, we and also the team from Helios have met with investigators in Japan who are tremendously enthusiastic and excited about getting this study up and running. And I think that when you have that degree of investigator enthusiasm, things can happen very quickly and efficiently. I think investigator enthusiasm, as I'm sure many of the people in the audience can relate to, that is actually a key ingredient for getting studies done efficiently and effectively, and that gets you to the data much sooner. And I think we're excited about that, not only in Japan, but outside of Japan as well.
I remember, like it was yesterday, the day that Clevidine from Pharmasset had failed. Pharmasset's market cap fell to 70 million. They had 20 million in cash, and they had about 15 employees in Princeton, New Jersey. And I remember getting the call from Schaefer Price saying, that they had this nucleoside for hepatitis C, and he was frustrated in the value. We all know a few years later that Pharmasset was acquired for $11 billion. So I would say to both of you, it's, I know it's very hard, but when you look at the stock price and you look at what's happened to regenerative medicine, but I do believe the pendulum's going to swing, and what's going to swing it is going to be data. It's just absolutely got to be data driven. With that, I want to transition to Dr. Jason McCarthy to introduce Jeff Walsh from Bluebird and start us off on the gene therapy side of the panel.
Thank you, Jason. And as Jason Kolbert was saying before, there was a time when phase 2 data was just used to really design and map out your phase 3 trial, and that paradigm seems to have been disrupted in gene therapy, because last spring we saw great data in beta thalassemia from Bluebird, but as Bluebird's stock was approaching and almost going through $200 last June, we got introduced to that N=1 story, where they essentially treated one sickle cell patient. Everybody pretty much knows the story, and the patient remained transfusion free. And as we headed into ASH, and when we came out of ASH, we saw data from 3 more patients, 2 were evaluable, and the data on the surface was not as impressive. It was very early on, and the Wall Street reacted very negatively to that. So just with 3 patients, the stock had swung from $200, and it's now down around $50, which is really interesting, because Bluebird had never discussed publicly saying that the data that they have is definitive proof of concept, that it's going to take time, and it's probably more of a 2016, 2017 story. But anyhow, they are changing the landscape in sickle cell disease. And here to discuss and give us an update is Jeff Walsh, Chief Operating Officer. And Jeff, if you could discuss with us, the catalyst and the data that we are going to see in 2016, because, in my opinion, it is a pretty big year for bluebird and sickle cell. I think we'll see a lot more patients, and also in immune oncology. And we're going to also see some data from the much less spoken about adrenoleukodystrophy, which I think is really interesting.
First of all, thanks for letting me relive in a few seconds 2015. That was a lot of fun. I would like to say, though, that there just to your point, Jason, it's been really fascinating for us to watch not just bluebird, but the field mature. And it started out as lots of questions about safety, and that was the first hurdle to overcome. And then it's proof of concept, whatever that means, proof of concept clinically. And now you're moving and you're seeing a lot of emphasis on robust data. More patients. Does it translate to a broader set of patients? And then increasingly, you're starting to get questions about commercialization. So I think that's the evolution that companies, us and others, are going to go through. And the reaction from the street is going to be the reaction from the street, but the long-term perspective always has to be kept in mind. That's certainly our perspective, and we've always been true to that we're going to release the data as the data come, and it's going to mature, and you'll see it mature accordingly. So it has been a very fun ride in 2015, and we're excited about 2016. I would say we really have 4 programs, really 1 or 2 of which were significantly highlighted in 2015. That's the beta thalassemia program and the sickle cell program that got all of the attention. But people do forget about our adrenoleukodystrophy program, partly because we haven't disclosed data, because we believe we're in a potential pivotal trial there, and you obviously have to be more careful about releasing data publicly in that regard. So we have 2 programs that we believe we have shown the reason to believe, whether you call that clinical proof of concept or not, where we have had amazingly transformative data in at least 1, and then obviously in another case, many patients. So I'll talk for a second about how we're going to talk about those programs. That's the beta thalassemia program and the sickle cell program. But we have 2 other programs that you could argue have pseudo-POC, in that we had a prior product in ALD that showed some pretty interesting data, and now we're transferring that clinical program to a new, hopefully improved program or product. And then we also have our BCMA immuno-oncology program targeting BCMA, as I just mentioned. And for that, at ASH, NIH had a program where they disclosed data targeting BCMA, where they effectively, I believe, validated at least the target is an interesting target to pursue, within the highest dose group getting 1 complete response and 1 partial response. So that's really exciting for the BCMA target as a whole, and I think you're starting to see a lot of discussion about BCMA as one of the next, increasingly next exciting targets for the CAR-T world. As it relates to 2016, in terms of clinical data and then also starting of trials, we plan to, on the BCMA program, have our first patient in the first half of this year. And it's a classic 3x3 clinical program. So depending on how that dose escalation goes, will determine whether there's any data to disclose. But really, we're anticipating that's more of a 2017 event. We also, in the first half, will, and we've submitted for, so hopefully will be accepted, will likely be presenting data on our ALD program for the first time with the new product. It's terribly exciting for us, and obviously for the patients that are on the therapy and that have the hope of this therapy working for them, hopefully in the future. So we'll give an interim update at AAN in April. That's the first time, again, we'll disclose data on the ALD program. And then in the second half of the year, the start of our 207 trial, which is the next thalassemia trial, focused only on non-beta 0 population. And then at ASH, we will be giving a more robust and full update on both our sickle cell programs as well as any of the beta thalassemia data that has emerged from any of the ongoing clinical trials. So that's what we expect to see in 2016.
All right, and maybe you can just give us a minute on sickle cell disease. Seems to be a very special disease. There's new therapeutics coming out, and where gene therapy is going to fit into that treatment paradigm as it's becoming more and more of a reality that it is going to be an option. We already know that transplant, sibling to sibling, works. Where that falls and what that market opportunity could look like.
Yeah, I'm not going to fully speak to the market opportunity. I think we all know the data in terms of number of patients that are out there. It's a fairly well-studied group of population in particular, at least in the U.S. and Europe. On this one, as was just stated, it's really data dependent on the programs. And we believe with the first patient in our 205 trial, we've shown that this can work. It's 6 months of data, so we all have to be careful about the longevity of data that we have. But we have 6 months of data that looks like it's having a pretty dramatic effect on a sickle cell patient for the first time that gene therapy has been applied to a patient. What we need to do internally is to be able to show that in more patients. And that's really what our 206 trial is all about.
Okay, great. And I want to thank you, Jeff. I want to shift gears to gene therapy for ophthalmology indications. And we have Applied Genetics Technology, CEO Sue Washer here. The company is very active in XLRS, and I'll let her give you a brief description of that. And the chromatopsia, they recently did sign, this past summer, a deal with Biogen that's potentially worth over $1 billion. And again, as Peter was describing earlier, it's another example of how big pharma is starting to recognize gene therapy. And here to give us an update is Sue Washer.
Well, thank you, Jason, and thank you for having me on the panel today. Certainly, AAV gene therapy and gene therapy in ophthalmology has become a hot topic over the last 12 to 18 to 24 months. There's more and more interest in the space. And while ophthalmology indications are not life-threatening, they are, sight is really the most important of the senses to human beings. Many people report that they fear losing their sight more than they fear death. So it's a really life-changing event that can happen to someone to be legally blind or to be severely vision impaired. We're very excited for 2016 because, as Jeff said, we're really focused on the long term. We want to get people focused on bodies of data, not patient-by-patient data. I would challenge anybody to say, even in the antibiotic space, the monoclonal antibody space, and even pain relief space, if you started looking at the data patient by patient and trying to make your investment decision in that way, you'd probably miss on a lot. So we're trying to focus people on the long-term data and a larger body of data. And for that reason, we changed our guidance earlier in 2015 to say that that's indeed what we were going to do. We were going to release data as robust packets of data over several patients, several time points, so you really can make that careful analysis and interpretation. And we're going to be able to do that in 2016 over 2 different indications, X-linked retinoschisis, which is a disease that causes very poor vision as well as a very fragile eye that can have retinal hemorrhaging and detachments, and about 35,000 patients in the US and Europe, and so we'll have data on that in 2016. In addition, we'll have data on a chromatopsia caused by defects in the CNGB3 gene, which is a gene that allows the phototransduction cascade to be initiated in the eye. These patients are all legally blind, and they're extremely photosensitive. In this room, they'd have on very dark tinted glasses because of that photosensitivity, and so we'll have data on that program as well. In addition, because of the partnership with Biogen, because of the finances we've been put in place, because of the really robust intellectual property partnerships that we've been able to execute on, and the staff that we have on board, we will be able to bring forward 2 additional programs in 2016 and early 2017. Another program in a chromatopsia caused by a second genetic defect, and a program in X-linked retinitis pigmentosa. So really, I think the message that we have is that we are looking at the long term. We're looking at the robustness of the data. We have the resources on board to collect these 4 sets of data in ophthalmology across very different diseases with a couple of different delivery techniques. So we'll be able to really look at a body of data and be able to make decisions about how best to move the programs forward.
And how many patients, before we do you think that we need to see? It's definitely, it seems to be not an N equals 1 story in gene therapy, as some investors believed early in 2015, in X-linked retinoschisis and achromatopsia. How many patients do you need to see positive data and to be suggestive of proof of concept?
Well, I think as Jeff mentioned earlier, most of these studies are dose escalation. So they're like 3x3, low, medium, high dose. That's what all of our studies are set up to be. And so I think what we're looking for in the beginning is there a dose response? First and foremost, safety, obviously. But then is there a dose response as you go up that curve? And I think that while we in the early dosing may not see complete or final or achievable full clinical response, we're looking for that trend over the dose response curve. In addition, all of our trials are set up to have to be able to immediately roll into an expansion cohort. So if we find a dose that's very promising with no regulatory interaction really other than the Data Safety and Monitoring Board, we can roll right into that expansion cohort and be able to develop that real proof of concept data at a dose that's clinically meaningful. And so we think that flexibility that you can build into these trials allows you to very efficiently get that body of data.
Great, thank you. So now I want to turn it back to Jason Kolbert, who's going to introduce our last guest.
Well, I asked Dr. McCarthy to give me the privilege to introduce Dr. Sven Keeley. Sven, I'm very excited to have you on the panel. You're the only person I've met that can talk about both sides of this equation, both gene therapy and regenerative medicine. For those of you who don't know Dr. Keeley, you would probably be fascinated to know that he was instrumentally involved in products like Macy and the Genzyme, in some of the Genzyme cell therapies. He lived through Sanofi Aventis. He watched the divestiture of that business to Vericell, and then made the decision to go back to big pharma, be the head of gene therapy at Glaxo. But I believe in Sven's heart, he really is looking for the pendulum to swing back to regenerative medicine. So what I really would like you to talk a little bit about is partly your decision to be the head of gene therapy at Glaxo and what is Glaxo working on, but also, you've got a lot of interesting experience in how big pharma has viewed cell therapy. It's been a little bit of a boom and a bust. Do you expect that pendulum really will swing back? What is Glaxo thinking, not just on gene therapy, but I know that at every chance you get, you probably talk to them about the regen side too. So close with us a little bit about how we can look at both sides of this coin that involves both the gene side and the regen side of the cell therapy space.
Well, thanks very much, Jason. I feel slightly under pressure here because you built me up so amazingly. So first of all, thank you very much for the invitation to come and speak today. I think where we're approaching right now, first of all, talking just a little bit about GSK and our emphasis, and I think GSK has been a little bit of a sleeper in terms of the cell and gene therapy environment for the last few years, and it's just recently we've started coming out, and I think we sort of came out with the announcement and with the filing at the middle of last year for the ADA SCID indication, which is probably the first ex vivo gene therapy to be filed with the European Medicines Agency. And we're very, very excited about that, and we're expecting to have a response from the European Medicines Agency in the first half of this year. So I think that's really, for me, one of the indicators that GSK has really taken that forward-looking step and thought that this is a space that really has a lot of potential in the future, and that this is really a space that's going to grow. So in terms of the work that we have on, we have a partnership with the Telethon Institute of Gene Therapy and the Hospital San Raffaele in Italy. And with them, we have a partnership for 7 different indications. Of those 7, as I've already mentioned, 1 has already been filed, 2 are in final pivotal trial clinical stages, and some of the early data has already been published on those, and those will be coming up very soon. And then we have another one which is already in clinical trials, and then we have another 3 which we're still in discussion about. So, GSK as a company has really decided to look at this area and invest very substantially, not only in people, but also in technology and the ability to move these products forward. I think most of you are probably also aware of the association that we have with Adaptimmune, and so that brings in the cell therapy side of it. And so from this you can see that the gene therapy and cell therapies are slowly starting to move together, and in my heart, I believe in the very near future we're going to be talking about regenerative medicine as this whole group, and there's not going to be such a big difference between cell therapies and gene therapies. At the end of the day, there's not a huge amount of difference in terms of how you develop them. There is some difference in how you manufacture them, but at the end of the day, these are still living medicines that require a very specific way of manufacturing, very specific way of showing safety and efficacy. And we've heard some great points today and some great news about the safety and efficacy that we're starting to see come out of this area. And so I think there's going to be a lot of convergence that in the very near future, when we talk about regenerative medicine, it's going to be cell therapy, gene therapy, and even tissue engineering all brought together. So I think those days of trying to separate them out are slowly coming to an end. I think in terms of where Big Pharma is coming, obviously I cannot talk about all the Big Pharma companies and what Big Pharma is looking for, because that would be completely inappropriate. But in general, in my limited experience, the reason that I went back to a large pharma company after some previous experiences, I swore I would never touch large pharma with a sterile barge pole. And what really got me back into GSK was speaking to the leadership and really talking to them and understanding from them this incredible belief that they see cell and gene therapy as the next pillar. If you think, for example, small molecules, and then you think of biopharms, and they really see that the cell and gene therapy is the next pillar, the next wave of being able to provide therapies to patients. And these are truly life-changing therapies and very often life-saving therapies. And what I saw in them was this real commitment to develop this area and to grow for patients and develop these therapies for patients that, let's be quite honest, as we've already seen from a lot of the failures around us, is pretty hard work for companies, because we're taking therapies that are cells that no one's used to dealing with. We're trying to show safety and efficacy with endpoints that are often 20 years in the future, and we're trying to get a marketing authorization from regulators that are really worried about looking at population mechanics. And we're going to them with 5s and 10s of patients, and we're saying, da-da, there you go, it works on these 5 patients. Now give us a license to go out and treat everyone. And they're naturally sitting there and thinking, well, hang on a moment, it doesn't really work like this. Haven't we been here before? Haven't we been here with other drugs that have caused catastrophic side effects in the 60s and 70s? And so they naturally are very, very nervous. Also, they've developed over all of the years with small molecules, they've developed a very good recipe for how you show preclinical or non-clinical data, how you show clinical data. And here all of a sudden we're going in and we're saying, well, if we've got an autologous product, showing overdose doesn't work. And if we're giving it into a joint, well, you can't really talk about reproductive toxicity and things like that. And we're going in and we're challenging them and we're putting them very much off balance. And so this is creating a lot of tension and a lot of nervousness. So for a company, it's very, very challenging. And then just to make it a little more interesting, we're developing some of these therapies that are not going to be a tablet or an injection every day or every week or every month or very often every year. Some of these therapies, the gene therapies that we have, are potentially lifelong therapies. You are going in and you are changing someone's genome potentially for the rest of their lives. What do you do if they have a major adverse event 10 years down the line? How do you go in and reverse it? And so there are a lot of really big theoretical discussions that these regulators are having. And so for a company, it's very challenging for you to go and to reassure them that actually everything is okay. This is not as scary as it might seem. And then I think the last thing, which Edward touched on earlier, is the whole thing about value and payers and reimbursement. And here we're talking about particularly gene therapies and also the cell therapies that we're now looking at. These are long-term therapies. And how do you cost those? How do you show value for those? And those are some very interesting discussions that I'm sure GSK is not the only one having with potential payers. I think everyone here sitting on this panel is already having those discussions with payers. And those are some very, very interesting discussions. So I think we have come a huge amount of distance, and we've really come a long way. And I think a lot of the magic, and I say this in a slightly different way, the unrealistic magic is gone, and we've come down to the realism. And it's not, as one of my old profs used to say when we did an operation, you do it like this, bing, bang, boom, and voila, patient's happy. It's not like that anymore. The voila is gone. It's now down to the data, and we're seeing this from everyone that's discussing today. It's down to the data. It's down to being able to convince not only the regulators, but also the investors as to what we have, that this isn't the voila. This is the data that supports it, that makes a huge difference in patients' lives. And for those of you that are looking to work with Big Pharma and to really come into the area, I would say, once again, data is king. A great idea is fantastic. Passion is wonderful. Most companies will buy into a strong management team, but that only gets you so far. After that, you need to have data, and you need to have a good plan. You need to be able to go into those discussions and say, this is how far we've gone, this is the data we've got, but this is how far we're going to go. This is the plan that we have for going forward. And I'm fortunate enough to be in a lot of those types of discussions, and very often a small startup has only thought to maybe stay phase 2 and nothing further on from there. And so that's really important to think the long term. And just remember, getting an approval is just the first step. Getting paid for it, that's where the challenge starts.
Sven, thank you. I'm so glad that you were our last speaker, because that's exactly the theme that I want to close on, which is data. As a research analyst that's following this industry, I see dramatic differences between the panel we're having today and the panel we had last year. The one theme that you have to walk away from this panel, regardless of which company we're talking about, is data. It's the timing, the catalyst, the data, and how meaningful that data is going to be. We were not having these discussions a year ago. Just a year ago, we weren't talking about this. This industry is now rapidly maturing to the point that we're going to have data, definitive data that will answer the question, does it work? And of course, every time there's a clinical failure, within the seeds of that failure is the next success. We see that time and time again. I should remind all of you that Avastin failed a clinical trial before it was approved and became successful. So I do believe that regenerative medicine and gene therapy has great things in store over the coming decade. And I do think that our job, my job as a research analyst, and Dr. McCarthy's job, is really to kind of pick those companies who we think are best positioned to produce great data based on all of the facts that we have to date. And we're seeing a little bit of a separation in the industry. We're starting to see now some of the winners and some of the companies in the lead, and I think you're seeing a great example of that on the panel. So we're very excited to be here, and we're even more excited to see what kind of discussion we're having at this panel next year. Thank you very much for all of our panelists.